The floor is yours.
Thank you very much. Thank you very much for the invite, Basis. It's much appreciated. Nice to always get out and about and spread the word about the benefits of sleep. Just to give a little context, I came to the topic from a background of sleep-dependent memory consolidation more broadly, but obviously also with an interest in mental health. When we looked into the role, particularly of the consolidation, i.e. the strengthening of emotional memory during sleep, there seemed to be some sort of link with depression, but this really hadn't been investigated. So this was a topic that we've looked at a fair bit. A lot of this is worked up by my former PhD student, Marcus Harrington, who's now at UEA, and also a former master's student, Jen Johnson, who now works for the civil service, so much better paid than I am. So putting all of this together, I hope to give you a comprehensive sort of overview, if you like, of the state of the art where we are, which is not necessarily all that far along, but certainly a lot further than we were before we started. So I will obviously talk to you a little bit about sleep in the first place, because I obviously some of you know more about sleep than others, so I'll just assume that you don't know that much and just give you a very brief overview. And similarly then talk about sleep and emotional memory consolidation, i.e. how sleep helps to strengthen your memory for emotional things and why that takes place and what aspects of sleep might be involved. Before then talk about the relationship between sleep and depression, and then putting it all together in what we call the effect tagging and consolidation model, which is where we see how sleep, depression and emotional memory consolidation are all related to each other. And finally, what we might be able to do about that in terms of helping changing sleep to improve the symptoms of depression. Let's see. So you get different types of sleep during the night. Sleep is not just one thing. The different types we call sleep stages. And broadly speaking, for mammals and birds, you can have either REM sleep, REM standing for rapid eye movement. So if you've ever seen somebody sleep, you might see their eyes moving, and non-REM sleep, where you don't get this. By contrast, insects and reptiles are purely non-REM sleep. So at some point, REM sleep evolved. Within humans, non-REM sleep gets further divided up into different stages, of which the one that we call stage 2 is your classic light sleep, and you get this 40–45% of the night. I'm not going to talk any more about stage 2 today because we're not really interested in that. The really deep sleep, which is the stuff you really want if you're feeling excessively tired, and you will get, this is what we call homeostatic sleep pressure that leads to you needing this, is called slow wave sleep. This is non-REM stage 3, looks a bit like this. So really synchronized firing of millions of neurons during that stage. By contrast, you can see stage 2, higher frequency, lower amplitude, the light sleep. And then finally, REM sleep, which looks a little bit like this, which is quite similar, as you can see, to stage 1, which is just a little light transitional stage. And REM sleep, which is what we call paradoxical sleep, or sometimes it still is, has been called so because it also has some similarity to a waking EEG pattern, and it's like being awake while being asleep in some sense. REM sleep is when you get your most vivid dreaming, though obviously you do dream during other sleep stages as well. So REM sleep, paradoxical sleep, similar, as I say, in some ways to being awake. The striking thing about REM sleep, so this background EEG is very sort of mixed frequency, but strongly powered by theta rhythms, so 4 to 8 hertz, if that makes sense. If you know about spectral power and analysis. So you have 4 to 8 cycles a second on average, with strongest power, and you also get these 2 eye channels, so lots of actual eye movements. So these are quite distinct, striking eye movements. The eye movements you get in REM sleep are quite different to eye movements anywhere else. So they're very distinctive. You can even tell them apart from waking eye movements. REM sleep is definitely involved, and I will show you some evidence for this later, in emotional memory consolidation. So we know that REM sleep is very much about emotion. Your amygdala, part of the brain which is involved in emotional arousal, can be up to twice as active during REM sleep as it is even during the day at its most extreme points. So you get really very strong emotional arousal during REM sleep. There is some evidence also of motor learning or procedural memory consolidation during REM sleep, but this is not really relevant for us today. And then slow wave sleep is the other one of interest, and this is, as I say, the really deep sleep characterized obviously by these slow waves. The slow waves play a coordinating role, so they coordinate different parts of the brain to be active at the same time. So you get reactivation in the hippocampus, called these little hippocampal sharp waves, and you get spindles in the cortex. And these happen at the same time, courtesy of the slow waves, which are coordinating them. And the idea is that there is a certain relearning going on. This is consolidation where memories are moving around. Again, it's not something I'm going to talk much more about today, but just so you can see where it is. I'm showing you slow wave sleep because in our stimulation later on, I'll show you what happens if you stimulate at a slow wave frequency versus a REM frequency. So slow wave sleep is characterized by very strong delta power. So 1 to 4 hertz, and especially the slow wave power 1 to 2 hertz region is really very strong, because obviously that reflects these slow waves. So that's sleep in a very short time. Now, emotional memory consolidation is something that takes place during sleep. Neutral memory consolidation is also something that takes place during sleep, and there does seem to be a distinction, as we will see. So we know that neutral memory consolidation seems to benefit from slow wave sleep. There's a lot of evidence about that. There is a fair amount of evidence now that emotional memory, by contrast, benefits from REM sleep. One of the ways you can look at this is to do a split night study, which we have here. So essentially, you want the two groups, if you want to compare two groups, you want them to have approximately the same amount of sleep. Otherwise, the difference in sleep is a huge confounding design. But ideally, we want one group to have mostly slow wave sleep and one group to have mostly REM sleep. Fortunately, you get most of your slow wave sleep in the first half of the night, and you get most of your REM sleep in the second half of the night. So a really easy way to do this is to do what we call the split night study, where you have one group who sleep in the first half of the night and are then woken up, and another group who sleep in the second half of the night and get to wake in the first part. So they get the same amount of sleep overall, but you can see just from the sleep parameters the difference between them. So the late people are getting only about 10% 10% slow wave sleep versus 28% for the early people. By contrast, the early people are only getting about 10% REM compared to 31% for the late people. So there's a really striking difference. So the late sleep is the REM-rich sleep, the early sleep is the slow wave sleep. In a study that was carried out, there have been many other studies using many other different types of stimuli, but this one was just one of the earlier ones by Wagner et al. They were using emotional text. So the text contains emotional and neutral words. So they have an emotional text group and a neutral text group. And they are testing content memory through looking for particular memory of keywords in their subsequent test. And they were comparing an early night group, slow wave sleep group, with a late night group, i.e. a REM group. In the early night group, interestingly, there are no significant differences in terms of sleep versus wake. So they also had equivalent wake groups for the same periods of time for either emotional or neutral text. So the slow wave sleep-rich sleep didn't seem to make any difference. By contrast, the late night sleep group, as you can see, the emotional text is much better remembered by the sleep group compared to the wake group. So this late night sleep really does seem to enhance memory selectively, particularly for the emotional items. So it clearly suggests the involvement of REM sleep in emotional memory rather than slow wave sleep or anything else. Just a little interesting aside, they did a surprise test on their participants, an astonishing 4 years later, which is quite remarkable, and actually you still see this difference in the emotional texts. So people who slept after having learned this 4 years earlier still had a better memory of the emotional texts compared to the people who stayed awake, which is really quite remarkable. Now, one prevailing theory around emotional memory and sleep is actually that sleep helps to strip out the emotion. At least this has been suggested as a theory. It's a very controversial theory. So the idea being that because sleep, and REM sleep in particular, is very rich in cholinergic activity, so acetylcholine is very strongly active during REM sleep, and that is obviously associated with memory, while the aminergic system, which is more associated with wake, is actually not strongly active during REM sleep, or indeed non-REM sleep, but it's also associated with more emotional things. So the idea being that during REM sleep you might remember these emotional events, but you might lose the emotional tone for them. At least this has been suggested. This is a Matt Walker theory, but not everybody accepts this. So one study to test this was done by Barron et al. And the key question on this study, so you've got a sleep group and a wake group, and then also just to control for circadian effects, just in case somebody is better in the morning or the evening. You've got a morning group and an evening group only. The key question here is, does the emotional valence of the stimuli change as a result of sleep or wake? So when you see it again after having had a sleep or having had a similar period of wakefulness, do you actually rate these same pictures, so this is images, do you rate them differently? The test is because obviously if sleep is really removing the emotion, then one ought to see a sleep-specific reduction in the emotional ratings. So, for example, the positive and negative items should move towards the neutral items. So, what do we actually find? Or what did they actually find, I should say? Well, the negative item ratings are very slightly reduced from one session to the next, so across the consolidation interval, while neutral is unchanged. But interestingly, the positive shift in valence for negative items, insofar as it exists, is more pronounced, you can see, for the wake group compared to the sleep group. So actually, sleep, far from helping to remove the emotion, if anything, is actually preserving the emotion, and particularly the negative, because these are negative versus neutral, so it's preserving the negative emotion of those items. You can also see this with the REM percentage, because people who had more REM sleep were more likely after sleep to rate things negatively, or their average rating was pushed actually even more negative for the people with the highest amount of REM sleep. So REM sleep seems to encourage you to see negative things as possibly even more negative, contrary to the theory. So that is emotional memory consolidation. Now we'll add a second component, which is depression, and then we'll put it all together. There is also a relationship between sleep and depression. We know there's an altered sleep structure in depression. In particular, REM sleep, which, as I've said, occurs mostly in the second half of the night. So there's a certain fairly long period of time for most people before you get your first episode of REM sleep. That latency is significantly reduced in people with depression. So people who experience depression get REM sleep earlier in the night. They also get more REM sleep overall, and that REM sleep is more densely filled with the actual rapid eye movements. Even more striking, perhaps, is the fact that these changes precede the onset of depression. Okay, so before you get depressive symptoms, you get these changes in sleep, and they are significantly predictive of depression to come. So they're actually a very good way of finding out who might develop depression as time develops. There are various reasons why this might be. Obviously, we see individuals at risk of depression show early REM onset when given a cholinergic stimulus. So you can actually encourage, you can pharmaceutically inject cholinergic or enhanced cholinergic activity in these individuals. And when you do so, you see that REM sleep occurs more rapidly, occurs earlier. And we know that the cholinergic system is very active in REM sleep. So, of course, this is perhaps not surprising. So this altered structure suggests that if you have depression, you are going to have more REM sleep and earlier REM sleep. And as we just saw, REM sleep may encourage negative ratings. So what about antidepressants? Well, one of the best documented features of antidepressants is that predominantly they reduce REM sleep. Not all, and the effects are quite variable, but you can see for the vast majority, there is a noticeable reduction in REM sleep. So MAOIs reduce it very profoundly, SSRIs similarly, fairly substantially, some of the others somewhat less. But it's reductions really almost across the board. It's only really NDRIs where this doesn't seem to happen, but potentially there might be an opposite effect. But for the vast majority of antidepressants, there is a significant reduction in REM sleep. Now, if we think that REM sleep is associated with depression, we know that people who have depression have more REM sleep. We know that people who have more REM sleep tend to rate things more negatively, and we know that people who take antidepressants, which may give them relief from their depression, also have reduced REM sleep. All of it would suggest that REM sleep is somehow, potentially through this enhancement of negative valence, is continuing to perpetuate the depressive symptoms once they have onset. This led us to develop what we call the affect, tagging and consolidation, or ATTACK model. So in a nutshell, we know that patients with depression have these changes to sleep architecture, i.e. there's more REM sleep. We know there's a reduction in REM sleep latency, and we know that the antidepressants reduce REM sleep. We also know that there is a strong perceptual bias towards negative items in individuals who suffer from depression, and there is stronger activation of the amygdala, i.e. stronger emotional reactivity to negative items in individuals who suffer from depression. And we know that that amygdala activity peaks during REM sleep. So in this sense, it all ties together. The model, in a nutshell, suggests that there is an interaction between these things. So people who are negatively biased, i.e. people suffering from depressive symptoms who show this negative bias, and this negative bias which has this strongly associated amygdala activity with it, have, unfortunately, more opportunity to consolidate these negative items because they also have more REM sleep. So it's this unhappy combination of being more prone to notice and remember negative items, together with having more REM sleep giving you the chance to remember these negative items, which leads potentially to a negative emotional memory bias. Now that's all in theory. Until our work on this, remarkably, nobody had actually tested this. It looks like it ought to be true, but is it really? So our very first study on this was to look purely at the effect of REM sleep on depression and emotional memory consolidation. So we were comparing individuals, this is based purely on their BDI score. BDI affects depression inventory, so this is a measure of depression. So we have low BDI group, these are essentially healthy controls. and the higher BDI group, so these are people with mild to moderate depression as evaluated by that. You can see the BDI scores average of around 3 for the low BDIs and up nearly 19 for the higher BDIs, so there is a striking difference between the two. They also differ inevitably really on anxiety. Wherever you see a difference in depression, you almost always see a difference in anxiety as well. So that's not surprising. That was the only other difference between the groups, however. So we wanted to do an emotional memory consolidation task with them to see if the individuals with depression really were remembering negative items more. So the task consists of, you see a crosshair originally, then you get the image for 750 milliseconds. Then, in the learning phase, all we want to know is an emotional valence rating, i.e. how positive or negative you perceive the image, and an emotional arousal, how emotionally arousing do you find it. The recognition phase is similar, except that in between the seeing the picture and the ratings, there is a memory task. And the memory uses what we call an RKN task. So this is remember, know, knew. So a remember response is a yes, I definitely saw this image in the previous set that we saw. So you clearly remember the item, you know when you saw it. The know one is a sense of familiarity. That looks familiar. I may have seen this before, but you can't necessarily place where you saw it. And then finally, the new ones are, no, I've definitely not seen this before. This is a new image. So this is a task that's very often used in memory studies because there does seem to be some evidence, at least for a neurobiological difference between the remember responses and the know responses. So the familiarity system may be a little bit different to the recollection system. We wanted to, in a sense, screen out the familiarity responses here and go specifically for recollection. So all of the results I'll show you are based purely on the remember responses here. These images are standardized images. They're taken from the International Affective Picture System, or IAPS. This is a database of images that have been rated for valence and arousal and other things like imageability. So they're widely used in emotional memory research. So that's why we chose to use those. So we have existing ratings for the images, so we were fairly confident that what we thought were negative images really were, but obviously we also have the participants' own ratings here to confirm. The design essentially is a split night study, same lines as Wagner. So again, we have an early sleep condition and a late sleep condition. And that's what we're really interested in. And each one, so during this 3-hour interval, they have an immediate recognition test, that's in the orange. And then after a period of sleep, during which we monitor their sleep. So in order to know how much REM sleep, how much slow wave sleep, etc. they get. Then they get woken up and they get, after a short delay to recover from sleep inertia, they get a delayed recognition test just to see what effect the interval of sleep has actually had on their memory. You can see from the sleep characteristics, again, it worked pretty much as expected. So if you look in the low BDI group, for example, the early sleep group, so these are the people rich in slow wave sleep, have 61 minutes of slow wave sleep versus the late group who only had 26. By contrast, the late group had 48 minutes of REM sleep compared to 15 in the early group. So this manipulation of early versus late sleep works very well in separating out slow wave and REM sleep. Other than those, and the number of rapid eye movements, which obviously relates to the amount of REM sleep we've got, all of the other sleep parameters were the same. So they got the same amount of overall sleep, same amount of non-REM stage 1, stage 2, etc. So the only difference here really is the amount of slow wave sleep versus the amount of REM sleep. So what we see in the low BDI group is nothing. So essentially, there's no difference between early and late sleep for positive, neutral, or negative items. So for the individuals who were not suffering from depression, there didn't seem to be any particular effect in terms of the valence of the images and their memory. By contrast, For the higher BDI group, so the depressed individuals, we see a very interesting interaction here, whereby the early sleep group are very good at remembering neutral images. So this top graph is essentially a measure of forgetting across the interval. So the lower the bar, the more the forgetting. So you can see the early sleep group has very little that has been forgotten, whilst the late sleep group have actually forgotten quite a lot of the neutral items. By contrast, the REM-rich late sleep group have remembered the negative items quite well and significantly better than the early sleep group. And also, as you can see, much more strongly than the low BDI group. So it does suggest that the individuals who are suffering from depression are remembering negative items in particular better than people who are not suffering from depression, and specifically during REM sleep. And more evidence of that is the fact that we get these individuals with the more rapid eye movements they get and the more dense their rapid eye movement sleep is, the more strongly they consolidate the negative items. So consolidation, better remembering of these negative pictures is specifically associated with the number of rapid eye movements. So this is pretty compelling evidence that the theory is broadly correct in the sense that REM sleep does definitely seem to be associated with negative memory consolidation. And it is specifically REM sleep rather than any other form of sleep. So what can we do to improve it? And there are a couple of things. So the first thing we did was we looked at sleep deprivation. So if REM sleep is doing bad things, it's leading you to remember these negative images while you're feeling depressed, which helps to keep you feeling depressed. One of the things you could do is to stop people getting the REM sleep. And the easiest way to stop people getting REM sleep is to stop them getting any sleep. So one remedy that is actually used occasionally for people with depression is total sleep deprivation. And there is literature on this showing total sleep deprivation is effective at reducing depressive symptoms for a substantial proportion of people for a period of time, though the period of time is rather controversial. And it certainly doesn't work for everybody, but it definitely does work for some people. And obviously the lack of REM sleep might be one way in which it works. So we actually decided to test this. So we have a total sleep deprivation group. They stayed awake completely all night for one night. We gave them an immediate recognition test. So we're looking at emotional memory to see, does this affect emotional memory consolidation? If we take their REM sleep away from them, by taking all their sleep away from them, is this going to change what they remember? And is that going to help? So we gave them then a recognition test 12 hours later. Obviously, 12 hours after sleep deprivation, you're in a pretty catastrophic state. So this isn't a terribly fair comparison. So we also did another follow-up test after 7 days. The task is similar again. It's a remember no new task. Again, using images. We slightly increased the duration of the images to 1,000 milliseconds just because it worked a little bit better. We have 180 images, etc. But it's basically the same task. So what we find this time is the greater retention of neutral and even more strongly negative items in depressed participants after one night of sleep. So if you look at the normal sleep group and then negative items, so this is a measure of consolidation for the depressed people, they're remembering them a lot. The people who are not depressed, you'll notice the opposite pattern. So the people who are not depressed, the low BDI group, are remembering positive items more and negative items least. The high BDI people are exactly the other way around. By contrast, however, the sleep deprived group, the black bars, you see, particularly if you look in the higher BDI group, they are not remembering negative items nearly as well as the people who slept. So it does seem to be helping them, and they're remembering positive images as well. So there is a significant three-way interaction effectively here, suggesting that the higher BDI group is different to the lower BDI group in terms of the interaction between whether or not you slept and So how well you remember stimuli of different emotions. So this does offer more support for the attack model and does explain, or at least give one mechanism as to how sleep deprivation might actually help. So it would help in this case by stopping you remembering those negative items when you're feeling depressed. Unfortunately, the lower two bars, you can see the effect after 7 days has pretty much vanished. So there are no significant differences anymore after 7 days. So this may work as a very short-term solution, but from our admittedly very small sample, this did not extend beyond even a week. Now there is a question as to why sleep deprivation does work for some people and does not work, really clearly doesn't work for other people. And so we were thinking about the genetic basis for this. Maybe there is a difference in genotypes. And there are 2 candidate genes that are of interest in this area, one of which, SLC6A4, is the serotonin transporter gene. So the serotonin transporter gene involved in actually the transport, obviously, of serotonin molecules across the synaptic cleft. So very strongly associated with depressive disorders, also known to modulate amygdala reactivity to emotional stimuli. So obviously highly relevant here, as well as obviously associated with things like fear learning. S allele carriers. So for the serotonin transporter gene, you can have either an S allele or an L allele, and the S allele carriers are the ones who are associated with higher emotional reactivity and greater susceptibility to depressive disorders. That's the short variant versus the long variant. And you get an approximately even distribution in our sample of the LLs, the LSs, and the SSs. So you get 3 groups. The other gene we were looking at was brain-derived neurotrophic factor, BDNF, and this is also associated with depressive symptoms. We know that this gene specifically is upregulated in the hippocampus during REM sleep, hippocampus involved in memory, REM sleep involved in emotional memory, so this is obviously potentially directly relevant, and there is some evidence that it might actually modulate REM density following emotional learning. So it's definitely worth looking at. This is a valine methionine job, so it's val versus val-met. The val carriers are the largest group in the population, and they were the largest group in our samples. So we have, fortunately, it's fairly skewed, and we have 25 val-vals, and then 11 what we're calling met carriers. So if one or both alleles is methionine, then we'll call them a met carrier. The met carriers, again, we're expecting to be more emotionally reactive and possibly with more REM sleep, and certainly more effective REM sleep. So what do we see? Well, first of all, there were no sleep differences between any of the groups. This is a good thing because we're not trying to change sleep between these groups, and we would rather it wasn't there as a confound. So that was fine. There, in the immediate recognition results, similarly, there are no differences. Again, this is fine because this is before any sleep, etc. So there shouldn't be any effect. Ideally, though potentially, obviously, there could have been because of the genetic variations, but there weren't in our sample, so that was good. After sleep, so in terms of delayed recognition performance, there wasn't an effect of the serotonin transporter gene, which was a slight surprise to us. You can see particularly the LS group is heading towards something which is pushing neutral down and putting negative up, but it was not statistically significant. A very similar pattern that was significant, however, was seen in the BDNF MET carriers. So the BDNF MET carriers are showing the strongest delayed recognition for the negative items and the weakest for the neutral items. So they are very strongly supportive of the role of BDNF, these results in terms of emotional memory, and in particular, the consolidation of negative memory. And you see these effects much more strongly here after sleep compared to nothing really before sleep. This is replicated in terms of the associations between the extent of the recognition performance and the REM sleep duration. So while the val carriers in BDNF show no association between memory performance and REM sleep duration. We do see a clear relationship in the MET carriers. So if you get more REM sleep in the MET carriers, you will, on average, be doing much better in your delayed recognition test. So in other words, if you're a MET carrier, you might see a REM-specific effect on your emotional memory that you don't see if you are a Val-Val individual. Similarly, we see this in the LS group for the serotonin transporter gene, which you'll recall was actually the group that showed similar overall patterns in terms of the recognition results as well. So we see an effect of REM sleep duration, but in specific genetic groups only. This might be why removing REM sleep has different effects across different individuals, because it seems to affect some genetic groups much more than others. What else can you do? So other than totally sleep depriving people, which is a bit drastic and not very nice for them, are there any other things that you could do to change the form of sleep they get and to help? So one other thing we tried was electrical brain stimulation. Obviously, all of these sleep stages are defined by EEG, i.e. the electrical brain activity defines what type of sleep you're in. So if you could alter the electrical brain activity, potentially you might alter the type of sleep you're experiencing. And therefore you might also experience changes of effects in this way. So this has been done in a number of ways. So transcranial magnetic stimulation, TMS, has been used. Transcranial direct and alternating current stimulation have been used. So this goes back to Lisa Marshall from 2006 as one of the earliest ones to do this. It's been done a few times since then. So you can see the typical pattern that there's learning, and then there's sleep, and during sleep there's stimulation during some sleep phase, some specific sleep phase. And the stimulation, as you can see in the grey part here, dominates the EEG signal whilst it's there. It's completely, that is the electrical signal. And then post-stimulation, there may be some entrainment effect or there may not. And that is a point that remains open to debate, and I'll show you our results just to give you an insight a little bit into that. So we did this actually with transcranial direct current stimulation, which is a very straightforward, remarkably sort of crudely straightforward technique in which you literally slap a couple of electrodes on particular places. One of them's an anode, one of them's a cathode, and then this electrical stimulation passes through whatever path in the brain, and it really is that non-specific. And he knows much more about this than I do, so he'll tell you much more about it. And that's pretty much what you get. So we did this, we decided to do this during REM sleep. So other people have mostly done it during slow wave sleep, but we were interested obviously in manipulating emotional memory and in particular manipulating REM sleep. Can you actually simulate REM sleep, and does this work? So we were stimulating for about 25 minutes during REM sleep. So we started in either the second period of REM sleep, and if necessary went into the third period, just because these periods are more stable than the first one and they occur later in the night. So we stimulated in one of three conditions. So there's a 0.75 hertz condition. So it's always with a kind of square wave signal, but it was occurring at either a 0.75 hertz frequency. This is very much a slow wave frequency. So this is effectively artificial slow wave sleep, if you like. We did it at 5 hertz. 5 hertz is very much in the theta range, so this is much more to enhance REM sleep. All of the stimulation is happening during or being initiated during episodes of REM. It's important to keep that in mind. So that's a REM frequency, and then also you should always do a sham frequency where there's a sort of stimulation that's not really stimulation, there's actually no electrical activity there. This is all within subjects, so we had 15 participants who did all three conditions. There was a cathodal electrode, an F4. There was some very tentative evidence from a previous study that this was involved in negative emotional memory consolidation in some ways. And obviously we were interested in that, so this is why we put that there. This simulation was done overnight, and obviously we were measuring polysomnography again while we were doing that. And we had our usual RKN task, only this time we decided to use emotional words rather than pictures. Not a particularly compelling reason for that, other than that we wanted to demonstrate you can do it. It's not just specific to pictures. And just as almost a little aside, as a control task, we also gave people what's called the serial reaction time task. This is a procedural implicit learning task. So basically a bunch of things light up and you have to click the corresponding key. And unknown to you, there is a specific sequence here. So you're learning, implicitly learning where how the sequence goes. So, and you can measure that by how fast someone clicks it, whether they're anticipating where it's going to be next. So this is seen very much as an implicit memory task, a procedural thing. So we put that in as a control. So what do we find? Well, in a nutshell, not a great deal, if I'm honest. So there was no difference at all between positive, neutral or negative memory performance in the sham condition, which is fair enough, but also in the 5 Hertz condition. So stimulating at a REM frequency during REM really didn't seem to make much difference to it. Curiously, sorry, that's supposed to say neutral, not negative. The 0.75 Hertz condition, we do see this boost here actually to neutral memory consolidation. 0.75 Hertz obviously is, as I say, artificial slow wave sleep, so this is a slow wave frequency. Slow wave sleep is very strongly implicated in the consolidation of neutral items. So in that sense, it's not surprising. It is, however, the first time it's been shown that you can stimulate in REM sleep at a slow wave frequency and still get the slow wave benefit. Previous studies had only ever done it during slow wave sleep, so it seems to work at any sleep state. So the slow wave stimulation works quite well, others unfortunately not. And there was no change, particularly in valence or arousal. So regrettably, this doesn't look all that promising as a method of removing or reducing emotional memory consolidation of negative items at all, either in healthy or presumably, we've not done it specifically in individuals with depression, but whether it would have an effect on those remains to be seen. Okay, so in a nutshell, we have this model, the effects tagging and consolidation model. Basically, people with depressive symptoms show a negative perception bias and greater emotional reactivity and activity in the amygdala, and they also have more and earlier REM sleep, and this REM sleep is associated with remembering those negative items. So they perceive more negative items and they have more opportunity to remember them. And that is the interaction that is in the model. So obviously greater retention of negative images in depressed individuals does seem to be specifically related to REM sleep, and even specifically to the number of rapid eye movements and the density of the rapid eye movements overall. If you deprive people of sleep, you can help some people at least, but it seems to be a very short-term effect, at least in our evidence. And one reason for this might be that there are different effects across the different genetic subgroups. So the BDNF MET allele carriers seem to show greater reactivity, and similarly, possibly, I'd say there's no effect on the serotonin transporter gene. That's half true and half not. The LS group do seem to show an association with REM sleep. They just didn't reach significance in terms of their group comparisons. Unfortunately, stimulating the brain at theta frequencies does not seem to help. Stimulating at 0.75 Hertz does enhance your neutral memory, but it doesn't make any difference to your emotional memory, so it's not really likely to help individuals who are suffering from depression. Future work, well, obviously we need larger sample sizes, so these are very quite difficult and certainly very labour-intensive studies to carry out. So it would be helpful to have larger sample sizes for some of these in the future. There are definitely different stimulation protocols that can be carried out and should be carried out. And it would be very interesting, and to my knowledge it's not been done at all, to test stimulation on individuals who are depressed versus not depressed, because there's been just no work at all comparing those at present. And it will be interesting to look at the effects of sleep deprivation versus antidepressant medications and various other methods. So TDCS is one, auditory closed loop stimulation, which is a different technique, but also to enhance slow wave sleep in particular is another possibility. Also REM specific disruption, so rather than whole night sleep deprivation, you could disrupt just REM sleep, and that might also show a benefit. So lots of stuff that could be done and should be done. So just finally, acknowledgements, Marcus Harrington, who did goodness knows how many nights in the sleep lab and other places to get a lot of this done, and similarly Jen Johnson and a few of my collaborators. And thank you very much for listening. Happy to take questions.